videos you're watching that week. You should always study the PowerPoint slides that are covered in those videos. And then in the corresponding content modules are optional Connect assignments that you're able to do to reinforce the content covered. The whole idea is by watching those videos, studying those slides, and potentially working on some optional practice questions, you're preparing yourself for the exams that are going to happen like every 4 weeks or so. Because the lecture portion is completely asynchronous, we never actually meet for lecture. The lecture exams are not proctored, so you're on your honor, etc., right? So only time we meet, right, is for lab. The good news is, because everyone in this lab is in that online lecture, it means that each week, if you wanted to, at the end of lab, ask lecture questions. I'm happy to sit down with you and go over lecture stuff, right? So the lecture is asynchronous, but at least you see me once a week so that we can like discuss, logistics, ask your questions, etc. All right, so let's go ahead and look into a module or two, get a sense of like what's there. So for example, in the course information module, I've got the syllabus for this class. I've got an information document with like more logistics, and then I've got like my A&P study tips. Let's just open the syllabus for a couple of moments just so we have a sense of like some of the major details there. Okay, it might look a little bit different as since yesterday. I just switched the syllabus onto the more accessible form where it's better for e-readers, for people with visual impairment, etc., but all the content is the same. All right, so if you ever need to get in touch with me, my office is like diagonally right across the hall over there, room 238. And because I'm a full-time faculty member here, it means that I'm on campus like at least 4 days a week. I've got office hours, which are scheduled, right? And notice though that my Monday office hour is online, but I might be here on campus anyway. So you could always, either see me in person or make a Zoom appointment, whatever works best with your schedule. Same thing for like the in-person office hours. If you'd rather meet in Zoom during those, that's fine because I can do that from my office computer. Of course, if my schedule of like office hours doesn't meet up with your schedule, don't hesitate to reach out. I'm sure we can figure out, a time to meet that works with both of our schedules. All right, a lot of the stuff on the syllabus is basic logistics, like the course description, right? Information about the course materials, some basic policies. One thing I'll point out in terms of policies, right? You're responsible for all material. Like if you miss a lab, you're still responsible for the material on that lab, right? If you're going to email me, please do so with your WCC email address. That way it doesn't get filtered out as spam. Obviously, no cheating, plagiarizing, using AI, etc. during exams, quizzes. And the tutorial center for science has moved out of this building officially into the ground floor of the library. So should you ever want extra help with lecture or lab stuff outside of what I can offer, you have the ability to get free tutoring there, so don't forget that that's an option. All right. I'm also, of course, in the syllabus supposed to put all like the objectives of the course, etc. But let's go ahead and take a quick look at grading. So just like A&P1, right, it is 2/3 lecture, 1/3 lab. So for the lecture, it is basically all exams, right? There are 4 unit exams throughout the course of the semester, and then there's a 2-part final exam. And separately, we'll look at like the lab syllabus to figure out how that 1/3 of the grade is determined. Just so you realize, every week is like planned out, right, for the whole semester, including when those online exams will be automatically deployed in Brightspace. So basically, it's like every 4 academic weeks or so, right, you'll notice that the week is bolded and there's a reference to an exam date, and those exam dates are always on a Friday. My thought process is that if a new academic week begins Monday morning, then if it's an exam week, the exam is at the end of that academic week, so the Friday. So that's the idea that each new week starts like new topics, and every 4 weeks, give or take, right, you take a unit exam. At the very end of the semester, there's going to be a 2-part final exam, but of course, right, that's 14 weeks from now. Cross that bridge when we come to it. So in terms of then how the Brightspace page for lecture is generally organized, there is simply enough a module for each topic. So, and inside each module is a pretty standard set of like topics or like posts, right? The first thing listed is always the slides for that chapter. After that are always the links to the various videos of me covering that chapter, in which I look terrible. Filmed during peak pandemic, lockdown, terrible haircut, in a sling from an arm surgery, looking great. I need to redo all of them just for that purpose. But anyway, gotta let it go. Okay, then underneath all those videos, you'll always find 2 optional assignments. No Connect assignments in lecture are required, even though they might show up in the electronic grade book in Brightspace. That doesn't mean it counts, it's just where you see numerically the feedback on the optional assignment. The idea is, a SmartBook assignment is where you read the textbook and it asks you questions along the way. If you get the questions right, you keep moving. If you get the questions wrong, it bounces you back. Some students love these because it forces them to analyze as they read, and other students are like, I don't have time for this, I'm not gonna do that, right? Up to you. Same thing with sets of practice questions. I went into Connect and chose every question that I thought reflected the level of detail that I cover in this course and built out these sets of practice questions. Again, they are recommended but not required, right? So you're free to work on them, help you study, but the score on them does not matter, right? I'm not looking at the grade book of these optional assignments. All right, and then again, they're currently hidden, but eventually, well actually no, I did open the first lecture exam, so that one's not hidden. Most of your exams for the semester are currently hidden, but since the first one is only a few weeks away, right, I made that one visible. So again, notice it'll be deployed like the whole day of that exam day, from like 7 in the morning till midnight. And it's 50 multiple choice questions with a 1-hour time limit, very similar to what you would have in person, right? So that's the idea of how lecture is gonna work, right? Each Monday, a new announcement will open telling you what videos to watch and like what slides to study. Throughout the week, you watch those videos, you study the slides, you potentially work on those optional Connect assignments, preparing for an exam that'll happen like a few weeks away. And nicely, you'll be seeing me in lab once a week as those exam dates approach, so we can always dedicate some lab time to your lecture questions, you know, should you like to ask them. All right, so anybody have any questions about the asynchronous lecture before we discuss lab logistics? I don't know if there's anything I might have left unclear. Well, even if we go on to discuss lab stuff, you can always jump in with lecture questions anytime. But just to remind you, you have a lab Brightspace page and a lecture Brightspace page. And because of that, that means we have a Connect lecture page and a Connect lab page. 4 pages. Okay. Thankfully though, right, Connect and Brightspace are integrated, right? So everything you would have linked in Connect is already here in Brightspace too, right? So there's kind of like 2 pages with 2 pieces, whatever. But anyway, so just make sure that on a weekly basis you're checking in with both. Brightspace only sends you email alerts if you tell it to do so. So therefore, if a new Monday announcement opens every Monday morning at 7 a.m. and you don't have Brightspace alerting you, then you might miss the announcement of the week. So either change your Brightspace settings to send you those alerts, or make sure that every Monday you're checking what the announcements of the week are telling you. Right? So for example, a typical lab announcement will tell you what lab we're covering that day, if there's a quiz to be taken, might remind you if there's any like virtual lab homeworks to do, etc. But just like lecture, every Monday morning, one of these will open up. Also like lecture, the course information, right, is where you find the syllabus, the safety document that you all just signed a little bit ago. For those that came in a little late, I'll get you those in a bit. But just to show you that the syllabus for lab looks just like the syllabus for lecture, the only differences are like the grading breakdown and the schedule. So in terms of grading here in lab, most of your lab grade is based on exams, And just like A and P1, exams include both the practicals where you walk around and more written-based like multiple choice exams. I also, though, deploy weekly quizzes, and those are going to happen in Brightspace. The exams are going to happen in person, but if I want to give weekly quizzes, I don't want to be spending 15, 20 minutes of every single week's lab for you just sitting there taking a quiz when I can instead deploy them electronically. So basically, every week, a quiz of the week will open up at like 6 or 7 in the morning, but will be due by 8 p.m. So you have pretty much the whole working day to get it done, right? Should be plenty of time. So you're never taking the weekly lab quizzes like here in lab, you're not being proctored for them, and that's why I keep them as a relatively small part of the grade, right? It's just a way for you to kind of make sure you're studying week by week, keeping you on target. The goal, of course, is to prepare you for those larger exam days, which we'll see on the calendar in just a minute. But then, a small portion of your lab grade is connect assignments called virtual labs. When the pandemic started and we had to go fully remote, right, that's how we were able to do hands-on things, right, electronically. And even though we're back in person, I now still utilize those as homework assignments. One, many of those virtual labs are where you're doing things that we couldn't do in this lab in the first place. For example, we don't have the materials for you to be doing a lab where you are like measuring the hemoglobin content of a person's blood. But you can very easily do that electronically in a simulation, right? And there's other things that we might do in these virtual labs that would be very expensive to have the materials here in person, so cool to just do them electronically as a simulation. So I'll show you those in a little bit, but they're, a small portion of your grade, 10%, not huge, but enough that you don't want to just throw those points away. I had a few students last semester in like my A&P1 lab who did 0 of these assignments, which meant the highest grade they could get in the lab was a 90, because they just threw away 10 points. I'm like, you paid for this class, right? Just throwing points away? Okay, just checking. Anywho, don't be that person. All right, and then the schedule. Okay, so here's how it works. Today we're covering lab 1 on the eye. What do you know? There are like virtual labs to do over the week ahead on the eye, and then next week is when I deploy the quiz on the eye. Next week's when we move on to the ear, right? And what do you know? What's the following week? Oh, the quiz on the ear. Right, it's always a quiz on the previous lab's content. Then, what's pretty nice is that before an exam day, there's always a review day. So, right, the first 3 weeks, we cover eye, ear, blood, and then we have a whole review day before the exam day. And that pattern continues. It's like we do blood vessels for heart, then blood vessels for 2 weeks, then a review day before a lab exam day. But what if a snow day happens between now and one of these exam days? Which days do you think would be extracted from our schedule? The review days, right? We're not going to cut content out of the semester. If we lose a lab day, we lose a review day. Hopefully it won't happen, but there was a spring, like maybe the year right before the pandemic, where it was Wednesday snow days the first 5 weeks in a row. Wednesday classes a couple of years ago didn't meet till a month in. It was crazy. It was just like pure chance. But anyway, hopefully that doesn't happen. But there was that semester where we lost all 3 review days and even 1 content day just by chance of snow. Right? So just be aware of that, right? Don't be like, oh, I'm going to skip this day because I'll just go back into lab on the review day and see everything. Well, maybe that review day will be canceled, right? Maybe not, but always want to be here, obviously. All right, and of course, the dates where practical and written exams, like the exam days, are clearly right on the syllabus, so very important that you're here. All right, so let's see how our typical modules are organized here for lab. So let's open up the first one, since that's what we're covering today anyway. Okay, the first link inside each lab module is always the notes of the day. So before you got here, or some of you got here a little bit ago, right? So if I didn't get you one of these, I've got them, there we are, right? Our notes of the day. Did anyone else need one that didn't get one yet? Oh, yeah. And here. Okay, so here's how these work. Every bit of outline in these documents are fair game for multiple choice questions on quizzes or exams. Then, as you look through, notice how certain names are underlined in the outline. And if a name of something is underlined, that's how you know, oh, this is going to be on the practical. And what I therefore do is at the end of the document is a list of all those underlined names, so you have an easy checklist of these are all the things I have to see on models or specimens or slides, right, wherever it may be. Okay, after that, there's usually just a PowerPoint show of diagrams. In a typical lab day, I kind of lecture my way through these notes, and while I do so, I project on the screen images that illustrate these notes. And you always have that PowerPoint file of diagrams where there's no notes, it's just images, but that way you have them to reference. Okay. Again, even though we are back to campus, right, post-pandemic, I still utilize the online electronic resources in Connect quite a bit. Part of that is because it brings in new ways of learning the same anatomy, which only furthers your understanding of it. So for example, in every lab module, I give literal click-by-click instructions of how to go into Connect and look at either like the virtual cadaver or virtual slides or watch interesting animations. And so every week I encourage you to like follow these instructions and kind of explore in Connect, because not only will it like reinforce your studying, but also the weekly quizzes that I deploy here in Brightspace can actually utilize screen grab images from Connect. So for example, today you're going to learn about the eye, right? And we've got plastic models of the eye, and even later in lab, I'm going to do a dissection of a sheep's eye. Well, in Connect, in our virtual cadaver app, we can like slice open a cadaver eye and highlight its different parts. In Connect, there's also virtual slides where I can like bring up a slide of a retina and highlight its different cells or layers. The thing is, I can then, from that app, literally just screen grab those like sliced eyes or screen grab those retina slides, and I give them to you in Brightspace and say, all right, this is part of what you should also be studying. So what I want you to notice is that in addition to the lab content modules, like lab 1, lab 2, etc., notice how there's a module toward the bottom called Screen Grab Images for Quizzes. That way, there's no surprise as to what images could show up as screen grabs on a quiz because you're given them in advance. But what you'll notice when I open this file up is that none of them are labeled. So part of your work each week is to theoretically be able to come in here and say, can I name all these things from the lesson I got from lab this week? So for example, as I scroll down, a lot of this is stuff we haven't done yet, blood, ear, etc. But I eventually get to like the virtual cadaver's eye. And there it is, right? And notice as I scroll through these pictures, different things are highlighted. So just to tell you, right, I can tell that's the cornea of this cadaver's eye, the clear window on the front. Whereas just below, oh, that's a picture, label, or highlighting a muscle of the eye. We'll learn today that that's the inferior oblique muscle right there. The thing is, that muscle, right, is literally exactly this muscle. The idea is, sure, I could say, well, I'm only going to test you on what's here in plastic in the room. But why would I do that when it's the same anatomy? And if you have to one day take a quiz with a cadaver and another day take an exam with plastic models, that means I'm forcing you to kind of learn the anatomy from multiple different angles, because it never should be about memorizing this model. It should be about knowing the anatomy in your head and then figuring out how the model or the image matches what you know. So the more variety I give you, the better you learn the anatomy. So hence the idea that throughout the week when you're in class, you want to be looking right often at the actual materials, but at home during the week, you want to be completing any virtual lab assignments and making sure that you can identify these screen grab images because they would be incorporated onto like the weekly quizzes. Realize you have the ability to download that file of screen grabs and add your labels. I am then happy to check your work. Numerous times students will email me a PDF and they'll be like, can you let me know if I got any of these wrong? And I will scroll through hundreds of slides checking your labels. And if you have 10 of them wrong, I will send you a reply being like, slide 32 should be this, slide 57 should be that. So in other words, if you put the work in to download and label every image, I'll match you and check it to make sure you're studying it accurately. So don't forget that's there. And in a typical lab week, I'll often bring us into the virtual cadaver as part of the lesson, so it's not like something you're only doing on your own. All right. Also, inside the lab modules, this time are going to be required assignments in Connect. I mentioned earlier in lecture, nothing in Connect is required, they're all optional. But as I showed you before, those virtual labs are 10% of your lab grade, so that's why these links to the various virtual labs are labeled as required. So hopefully that's pretty clear that these have to be done, unlike the ones in lecture that are optional. All right, and then finally, what do you know? At the bottom of this first lab module is the link to take next week's quiz already visible to you. It's a full week away, but at least you'll know next week where to go to click on it to begin it. So notice next Wednesday at like 7 a.m. it opens, and it only has a 25-minute time limit, right? So therefore, you could get to campus next week like a half hour before lab begins and take it here before 8 a.m. Or you can wait till after lab because it's not due till 8 p.m. Totally up to you, take it on your time. All right? So that's pretty much how the lab is going to work. Like every new lab day has a whole module, and in there are the various notes and links to go to Connect, any assignments, etc., right, and the quiz. All right, I think that's pretty much it in terms of like lab logistics. But of course, maybe I've left things unclear, so feel free to jump in with whatever questions you might have. And even if for the next couple of minutes we officially get into our lesson of the day on the eye, right, you can always jump in anytime with questions about like the course in general. Okay. So again, if I missed anyone for the packet of handouts of the day, the pile is up here. Help yourself if you need one. Otherwise, my thought is to officially open up the diagrams and get into the notes. Okay. So, our goal for today is to discuss the anatomy of the eye and the physiology of vision. And in terms of what I have as like Roman numeral 1 of our notes of the day, notice I'm calling it the accessory structures of the eye. So no surprise, right, our eyeball has structures that surround it, right? Muscles, eyelids, right? We have a gland for producing tears. All of those things that are outside the eyeball can be thought of as then accessory structures of the eye. And so hence I made a whole Roman numeral number 1 about those accessory structures. So first of all, when I was in college, I'm sure my professor would have insisted that we call our eyelids palpebrae, or a single one palpebra. But more recently, eyelid has become anatomically acceptable. I'm fine with that. The more common names we can use, the better. So sure, in this diagram, they're using inferior eyelid, superior eyelid. Yes, if on a practical, I am specifically putting a sticker on the upper or the lower, probably good to specify. But again, whether you call them an eyelid or a palpebra, either way is fine. What does the word fissure imply? Does that sound like it's describing one of these eyelids or the space between them if we use the phrase palpebral fissure? The space, right? We might have learned in A&P1 that a fissure is like a big separation, like what's in between the hemispheres of the cerebrum, for example. So yes, if I were to cut a sticker that kind of bridges this gap from upper to lower eyelid and said like name that gap of space, That literally is that palpebral fissure, and we see them kind of bracketing off in the diagram. All right. The next thing to mention is that our eyelids kind of meet at corners. So we can either call commissures or more commonly use the word canthus or the plural canthi. So for example, where am I pointing now? At my medial or lateral canthus of my eye? Lateral, right? Because it's away from the nose. Whereas over here, this corner would be the medial canthus, right? The corner toward the midline. Which canthus, the lateral or the medial, has a pink fleshy area, like kind of within that corner?
Medial.
The medial, right? When we look at the medial corner of our eye, near our nose, right, there's like a pink fleshy area in there. And quite literally, right, at your tables, right, are these plastic models with this removable piece. And that removable piece, right, shows that little pink fleshy area. That little area is known as your lacrimal caruncle. In there are glands like sweat glands, oil glands, right? And what's funny is, as we sleep, because our eyes are closed, those secretions then get stuck in that corner and tend to dry out. That's why in the morning we might sometimes wake up with some like schmutz or whatever, crusty, sandman, there's all these little nicknames, whatever, for like the stuff in your eye, right? But I had a student a number of years ago tell me they learned the word caruncle by saying, sometimes you wake up in the morning with a little juncle in your caruncle. I was like, that's a great way to remember it. But simply enough, right, that only that medial corner has that little fleshy area. Okay. Let's notice the next name listed. Levator palpebra superioris muscle. Just rolls off the tongue, name a dog that. Said no one. But what does it do? If it is a muscle called levator palpebra superioris, what does it do? What does it lift up or elevate? Your upper eyelid. Literally, while you're awake, right, your upper eyelids have to be held up. And there's a muscle in charge of holding up your upper eyelid. So notice on this plastic piece, is there clearly a red skeletal muscle that would be representing the muscle that grabs this upper eyelid? Yes, right? And that's exactly what that is. All right, so that is the idea of the levator palpebra superioris muscle, right, elevating the upper eyelid. You might have learned in A&P1, there's a muscle condition called myasthenia gravis disease. It's an autoimmune condition where people struggle to control their muscles. One of the diagnostic signs for it is drooping upper eyelids, right? If you have muscles that don't quite work, then these eye muscles that hold your eyelids up don't work. And so people with myasthenia gravis always look like they're falling asleep because their eyelids are like drooping. Just need to make that quick connection to something you might have learned about in A&P1. All right. The next thing is the conjunctiva. Notice I don't have the conjunctiva underlined, right? So you're never going to see it on a plastic model. But what term implies the inflammation of this conjunctiva? Conjunctivitis, right? Itis as a suffix on the end of anything implies like the inflammation of. What do we commonly call conjunctivitis? Yeah, pink eye, right? Okay, so if you're not really familiar, right, pink eye is a bacterial infection spread a lot amongst kids, right, because their immune systems aren't that strong yet, very contagious, etc. Right, but simply enough, your conjunctiva is a clear mucous membrane that covers over the front of the eye to protect it and prevent it from drying out. What's interesting though is a common misconception is to think that this slippery membrane that is the conjunctiva kind of wraps around the eyeball, as in the slippery membrane just literally like envelops the eye. No. Instead, the conjunctiva actually covers only the front of the eye and then folds to line the insides of the eyelids. That's pretty interesting if you think about it, because that means there's a continuous slippery membrane that runs from the inside of the upper eyelid, then down the front of the eye, then up the surface of the lower eyelid. So when you blink, is it your fleshy eyelid rubbing up against your eyeball, or is it slippery conjunctiva gliding over slippery conjunctiva? All conjunctiva. Because if there's a slippery membrane on the front of the eye, and the same slippery membrane folds to line the insides of the eyelids, then an eyelid sliding over the eyeball, right, is actually slippery membrane gliding over slippery membrane. And for that reason, if somebody wears contact lenses, could that lens get like lost behind their eye, or is there a pocket that it can't go beyond? There's a pocket, right, because the membrane folds, right? So that's the idea that the conjunctiva doesn't wrap around the eye, it creates a continuous membrane to cover only the front by touching the front of the eye and those inner surfaces of the eyelids. Okay, so because it's a clear membrane, that's why it's not underlined. I wouldn't ask it on a practical, you can't see it anyway, right? All right, but what about the term lacrimal apparatus? Does anybody remember where your lacrimal bones are? Here. Okay, for those that had me for A&P1, when we learned about the lacrimal bones, you might remember that I told you, you don't only cry out of your eyes, you also cry through your nose. What? I know. Okay, here is the idea. Each eye has a lacrimal gland, right, for producing tears. And the lacrimal gland is always found both superior and lateral to an eye. So quick question, is this model showing a right eye or a left eye if this lacrimal gland is always superior lateral to an eye? It's got to be a left eye. Because notice if I go to orient this to my right eye, the gland is near my nose, which can't be correct. So this must be a model of a left eye putting the gland to the lateral side. Very good. Okay. That gland then releases tears onto the surface of the eye, and then the blinking of the eyelids spreads those tears over to the medial side. I still remember as a kid, somebody like showing me these tiny little holes near their eyelids and being like, look at these that I found in the mirror. You think that's where the tears come out? And I was like, I don't know, probably. Well, no, it is not where tears come out of. It's where tears drain into. These tiny little holes in your eyelids, right by your nose, are actually entrances for excess tears to leave the eye surface and enter a series of tunnels. These two little tunnels, called lacrimal canals, brings us to a pouch called the lacrimal sac, which drains into your nose. Which is why, what do you hand someone when they're crying? A tissue. It's not because all of a sudden when a person's crying, they start making a bunch of mucus. It's because literally tears leave your eye and drain through your nose. That's why your nose runs when you cry, right? So that is the idea that these little green tubes on the plastic model represent these like lacrimal canals. And this green pouch that like gathers the tears, right, is this sac. So that's the idea that on this plastic model we see the gland that produces the tears and the tubes that like drain the tears. Pretty neat. All right, so that is the idea of the structures there for producing salty, delicious tears. Why are tears salty? They have to be isotonic, right, to our bodily fluids. Right, we learned in A&P1 that we don't want things to be hyper or hypotonic, right? So tears are naturally salty. Makes them delicious. Drink the tears of your enemies. Okay, don't actually do that. Anyway, all right, muscles. To finish out Roman numeral 1 on the accessory structures of the eye, notice I've got letter F called extrinsic eye muscles. Where do you think these muscles are if they're extrinsic? Yeah, they're on the outside. What if later we slice into the eye and we see structures made of muscle tissue in there that do things like controlling light levels or controlling focusing? What do you think we're going to call those tissues? Those are your intrinsic eye muscles. So very good, when you look at a plastic model of an eyeball, the muscles on the outside are extrinsic, but if we take the model apart and we see muscular structures within, then those are the intrinsic ones. All right. So, between the names oblique and rectus, which of those tells you eye attached to the eye on a 45-degree angle? Does that sound like rectus or oblique on being on a diagonal? Oblique. Right? You might remember from A and P1, your rectus abdominal muscles are on a vertical axis, but your oblique muscles fibers are on a diagonal. The word rectus, as in erect, as in like up and down, and the word oblique literally implies like on a diagonal. So here's what's then convenient. 4 of your 6 eye muscles are rectus muscles, and they not only tell you where the muscle is, but how the eye moves. So for example, what do you think you're going to call a rectus muscle on top of the eye? Inferior, superior, medial, lateral, what would you call it? Superior rectus muscle. And like the name implies, how does it move your eye? It makes you look up. Literally, when you pull on the top of the eye turns upward. So convenient that a superior rectus is not only superior to the eyeball, but it also makes you look upward. So what do you think an inferior rectus muscle makes the eye do? Look downward, right? So below the eye will be inferior rectus, right? And thankfully, right, it makes you look downward. What would you call a muscle here on the nose-facing side of the eyeball? Medial rectus muscle. And what about on the ear-facing side? Lateral rectus. Okay, here's the thing. I may never ask you, like I did before, is this a right eye or a left eye? But do you have to know if it's a right eye or a left eye, even if I never ask? Yes. Because I could ask you, which muscle is this? And if you don't know if it's a right eye or a left eye, then you don't know if it's medial side or lateral side. So keep that in mind when I've mentioned before about orienting right versus left, and I'll tell you other ways you can do that as well. Right? That's necessary even if I never ask that question, because you need an orientation to name the muscles. One more quick question. When a person looks up or looks down, they're using 2 superior rectus muscles or 2 inferior rectus muscles. But do you ever use 2 lateral rectus muscles at the same time? No, you can't move your eyes like simultaneously toward each ear. I can try. Did I do it? I don't think I did it. Okay, right? What happens when you look to your right? You're using 1 lateral rectus muscle and 1 medial rectus muscle, right? So that's the idea that these rectus muscles, right, move the eye in kind of simple straight line motions, right, up, down, right, left. But sometimes it's like, well, what's up there, like in the corner, right? Or what's down there toward that corner? That is where the oblique muscles come in, because they have the ability to make the eye move toward those diagonal corners. But here's what I want you to notice. Superior oblique is up here because it's above the eye. Inferior oblique down here because it's below the eye. But if you read the descriptions I put in your notes, or I can verbalize it for you, your superior oblique muscle makes the eye move down and to the corner. And your inferior oblique moves the eye up and to the corner. So are the oblique muscles moving the eye in a way that matches their name or is opposite of their name? Opposite. Let's see why that is. Notice how the rectus muscles insert on the front of the eyeball. Therefore, when they pull on the front of the eye is moving in that predictable direction. But the oblique muscles insertion is on the back of the eyeball. So if you pull the back of the eyeball down, where's the front of the eyeball going? Up. Right? Think of the eyeball as a seesaw. Whatever you do to the back, the front does the opposite. So if the oblique muscles grab the back of the eye, unlike the rectus muscles, then inevitably their movement is opposite their name. So that's the idea that the inferior oblique makes you look up by pulling the back of the eye down, right? Swiveling it upward. And superior oblique pulls up on the back of the eye, which is why it then makes you look downward. So that's the idea that the four rectus muscles, their names like match how they move the eye, but the two oblique muscles, their names are like opposite as to how they move the eye. Again, because the rectus ones grab the front of the eyeball, but the obliques grab the back. So everything is then like reversed. All right, hopefully that makes sense. And therefore, like just putting stickers right onto these muscles, asking you to name them, right, should be hopefully fairly easy. But quick question. Because the superior oblique muscle does this crazy loop through a pulley system, then what do you notice is on the top of the plastic model? The actual red of the muscle body, or only its white tendon? The white tendon. Right? Notice how there's this like looping thing that this muscle does, right? The muscle's tendon goes through a pulley system, if you will. But in this plastic model, it's like they cut the tendon right there, so you don't actually see the muscle, just this little piece of tendon. That little white piece of tendon is for the superior oblique muscle. We just don't see the red of the muscle, we just see its white tendon. Notice, that white tendon up there has been cut. Does that tendon point medially or laterally? Right? Like, is this going to go to the right eye or the left eye? Where does that tendon orient? To me, it looks like that little loop is near the nose, right? Because here's the lateral side of this eye in the diagram, and that little pulley loop looks like it's on the opposite side. I bring this up because sometimes students will ask, well, how do I know if it's a right eye or a left eye if I don't have the gland to go by? Well, no problem. The tendon for that superior oblique points toward the nose. So even if this part of the model is not there to show me that gland, no problem. I look at that white tendon and I know, oh, the nose must be over here. There's like another way to orient myself for right versus left. All right, hopefully that makes sense. Feel free to jump in, of course, if anything needs further clarification. But if I'm thinking correctly, that should wrap up Roman numeral 1, right? All these like accessory structures of the eye. And notice, right, numerous of those structures were on this removable piece. So no surprise, right, that plastic model like that could literally be its own station, right, on a practical exam. All right, so then here's the deal. Your Roman numeral 2 on our outline of notes of the day is where we officially start going through like the anatomy of the eyeball itself, right? I described the eye as a fluid-filled spherical structure. It's going to contain the photoreceptors needed to absorb light and send that information to the brain for processing. All of this is done in a 3-layered wall. So notice, in the big diagram, it literally looks like they have separated the 3 layers out from each other. And in the smaller inset picture above, they do an even more general idea of showing those 3 layers and labeling them. And what's convenient is that in that little inset picture, they have little boxes that list out which structures belong to which layer. So notice, the layers, like fibrous layer, vascular layer, right, they're not underlined. What is underlined are the structures that belong to those layers, like the sclera, the cornea, the iris. But do you think I would maybe write multiple choice questions where you have to know which structures belong to which layer? Of course I will. The questions practically write themselves. But anyway, let's go ahead and work our way from the outside to the inside, chipping away at these layers. Why would it make sense that the outermost layer of the eye should be the fibrous layer? What do you think all the collagen fibers that build that fibrous layer are providing? Yeah, protection, shape, support, right? The eyeball's got to hold its shape, keep in its fluids, and withstand the forces of these muscles tugging upon it. So yeah, the outermost layer of the eye better be strong to deal with all of that, providing shape, protection, and allowing movements by having muscles tug upon it. Okay, is the majority of that outermost layer white or clear? What do you think is the majority of that outer fibrous layer? The white part, right? Notice on the plastic model, when I hold it up and spin it around, right, the vast majority of what you're seeing is white. Many of the plastic models in the room have a missing piece. Don't mind, I'm just going to hold yours up for a second. Thank you so much. Notice how on this one, in addition to all the white, there's a clear window right on the front. Not all the models in the room have that because literally it just falls out. I literally precariously put them into position. Oh, look, I just removed the cornea. Just like that. Oh, I can put it back. Surgery. Anyway. But the idea is most of the outer, well, you get the idea. Held together by the best Scotch tape money can buy. Okay. They very precariously sit in there. But the idea simply is most of the surface layer of the eye is your white sclera. But the last thing you want is that thick white sclera in the front of the eye, because then no light would enter. So only in the front is that sclera swapped out by the clear cornea. Okay. So simply enough, all of the outer white surface of the eye, if I put a sticker on any white part of the eyeball and ask what is that layer, that is the sclera. But only at the front, right, does it get replaced by a clear window, and that's the cornea. So just to show you, at each of your tables, in addition to the three-dimensional plastic model, there's also this more two-dimensional sliced eye. And notice, just like this diagram here, most of the outer surface, like here, would be that white sclera, but only here at the front would be the clear window that is the cornea. Okay. Anyone know? If you get a little scratch to a cornea, is that going to be there forever, or does it grow out over time? A scratched cornea. It grows out. Why? Because your cornea is a stratified squamous epithelium. Where else do we find that? Your skin. Right? So your cornea, that surface layer covering of the front of the eye, the reason it can be regenerated, right, is because it's stratified squamous, but it's clear, unlike our skin. No pigment at all. Right? So it's clear to let light through, but it is stratified, which is good because that means if it gets a little scratch or anything like that, right, it just grows out. Just like the skin. All right, so those are the two major structures of the fibrous layer. The white sclera giving shape and support, and the clear cornea, right, allowing for light to enter. Okay, what is implied is found in the middle layer of the eyeball if the name of it is the vascular layer. Blood vessels, correct. In fact, the other two layers, the outer fibrous and the inner retina or sensory layer, are actually avascular. Why can they be that way? Because they are sandwiched and pressing against a middle layer that is full of blood vessels. So do the other outside and innermost layers need their own blood vessels? No, because they're just touching up against, right, a well-vascularized layer in the middle. That's why, logically, when you remove the outer white sclera of the model, what do you know? The next layer you see is filled with blood vessels. That's what we're seeing now. We're switching from the outer fibrous to the middle vascular layer. Okay. So, what you're seeing when you look then here is all of this dark part of that middle layer with the vessels is called the choroid. And then the colorful ring in the front here that gives you like your eye color is called the iris. And then if you turn to look on the inside of the now sliced open eye, you see a bunch of red hiding within, and that's the muscle called the ciliary body. So right, notice in the little box, it just seems that there are three distinct structures that belong to this vascular tunic, right? The choroid, which is the dark area with the vessels. The iris, which is the colorful ring that gives you your eye color. And the ciliary body is kind of inside, right? All the red musculature you see within. All right, let's discuss each of these three components of this vascular layer. Okay, the choroid, which is all this dark area with the blood vessels, tell me about things that are darkly pigmented. Does light tend to bounce off of dark pigments, or does light get absorbed into dark pigments? Good, absorbed. That is exactly what you want inside the eyeball. Why? Because you want the light from our stimulus around us to enter the eye and be absorbed and disappear. The last thing you want is for light to enter the eye and go boing, 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 like bouncing around inside the eyeball. Because then you'd be like seeing the same light over and over, and that would just make your vision a blurry mess. So you want the inside of an eyeball to be like pitch black. That way every bit of light that enters just disappears as it gets absorbed. So the choroid being vascularized, of course, gives nourishment, but also being highly pigmented and dark is adding to that darkness inside the eye we need to absorb light. All right. For whatever reason, students always struggle to identify the choroid. They get confused. So like whether it is an image on a quiz or a question here on a practical, right? A sticker right here on this brown layer filled with vessels, for some reason students struggle to understand it. But right, that is the choroid. The middle layer, nicely darkly pigmented, lots of vessels. Okay. So then notice, if we look at this middle layer from the front, right, anterior view, we see a colorful ring. Right? I might say this person has like blue-green eyes, right? But of course, that ring can be dark brown, hazel, some rare colors like lavender, purple, or gray, right, are in the human genome, right? You don't see those often, right? But that is your iris. What's interesting about the iris is that even though it can be colorful, it's actually made of muscle. We don't tend to think of muscles as being like green or blue, right? But in the iris, they can be. Anyone know what is the iris controlling the size of? The pupil. What is the pupil? Is it a tangible structure or is it a hole? It's a hole. That black dot right in the middle of your iris is a hole, as in light travels through there, right? The thing is, if it's a hole, why does it look like a black dot? Well, what do we say about the inside of the eye? It's extremely dark. So does light ever bounce off the back of the eye and come out of your pupil? No, light only goes in, never back out. That's why looking at a person's pupil looks like you're looking into like an abyss of darkness, right? Into their soul, right? Maybe not. Right? But the idea is that light doesn't reflect back at you. That's why it looks so dark and almost tangible. But it's not, right? It's an empty opening that light travels through. So if I look straight up at the lights on the ceiling, what should my iris do to that pupil? Dilate it, opening it up, or constrict it, close, like shrinking it down? Constrict, right? If I'm looking right at a light source, I don't want to wash out my retina with too much light, so hence constricting it. But what if we turn off all the lights, right, and we start to adjust to the darkness of the room? What would our eyes be doing to adjust like that? Dilate, right? Then you start to see better in the darkness, right, because you're letting more light in. And of course, fight or flight and rest and digest responses can also control those pupils, right? If you're running for your life from a predator, open those pupils, see everything so you don't trip, right? All right, so that is the idea of the iris, the colorful ring of muscle that controls the pupil opening to regulate light levels. I once had the most embarrassing moment because of eye adjusting to light levels. Might as well share it. When I was in college, I had a theater class where the only way into the classroom was a door directly from outside that went straight into the classroom and then was closed. So I, on a beautiful, bright, sunny day, arrived to class 10 minutes late, my own fault, and the professor was showing a video. So when I open the door from outside and it slams behind me, the room in front of me, pitch black. But everyone else had been sitting in there for the whole 10 minutes, so their eyes had all adjusted, so they could all see me, no problem. So the next 5 minutes was me doing this. Is there an open desk? Can someone point me to an open desk? Because I literally can't see it, and I'm feeling and everyone's just laughing. Oh my God, and I started laughing too. I'm like, thanks everyone. I must have looked so ridiculous. So don't do that. Anyway, be on time apparently is the moral of that story. Okay, so that is our iris. Okay, so now I skipped over the ciliary body. I saved that one for last here in the vascular layer because it's kind of the most difficult to wrap our heads around, but we got it. Let's discuss it now. So I'm going to do a quick drawing on the board because this image shows a sliced view, right? So notice here in the middle is something called the lens that we haven't discussed yet, but I'll mention right now that that lens helps to focus the light that we see. In this image, we are seeing a sliced view of it. On the board here, I'm going to draw it as if you're looking from the front. So now here's the lens right here. In this sliced view, I see little white strings above the lens and little white strings below the lens. So does that mean that the strings are only up here and down here, or are they actually all the way around because this is just a sliced view? They're all the way around, right? The two-dimensional slice, though, only captures what's on top and what's on bottom. All right, so there's the lens, and these little white strings are called suspensory ligaments. So it's almost like your lens is kind of hovering inside your eye because it is held in position by a ring of fibers holding it in place. Okay? Those suspensory ligaments then attach to muscle. This ring of muscle that I'm drawing now, which I'll even do this, kind of put like muscle fibers wrapping around. This is the musculature of the ciliary body. Okay. Question. Notice the ciliary body is a muscle, but it's a ring. What will the size of that ring do when the muscle contracts? It shrinks. Right? When a muscle is a circle and the fibers overlap more, which is what happens during contraction, then the circle doesn't get bigger, the circle gets smaller. Right? Because more fibers overlapping means the size of the ring muscle gets smaller. So what just happened to these strings? If the walls just moved in, did the strings get looser or tighter? The walls just moved in, which meant these got looser. How weird is that? A muscle contracts and the little tendons get looser? That feels backwards, right? Because usually when a muscle pulls, it tugs on its tendons, not loosens them. But if the muscle's a ring and the tendons go to something floating in the middle and the ring shrinks inward, then you didn't just tighten these strings, you just loosened them. So this lens is flexible. Did you just pull it flatter? Or by releasing it, did it just bulge thicker? It just got thicker. So the idea is, when you contract the ciliary body, you thicken the lens. So by contracting that muscle, that loosens those strings, and so the lens just got thicker. So what if you relax the ciliary body? Does that mean that we're moving the walls in or out? Out. So what happens to the tension on the little strings? They get tighter. And so what are we doing to the lens as we pull on it in all directions? Is it getting thicker or thinner? Thinner. Because you've pulled it tight, and that has thinned it. So again, hopefully we get the idea that when the ciliary body is in relaxation, then you end up making the lens get thinner. What are we doing by controlling that thickness of the lens like that? Is that changing how much light enters the eye, or are we using that to focus the light on the retina? Focus. The pupil size controls how much light enters the eye, but this changing of your lens thickness is how you focus, right? We'll learn about it later, but here's the idea. Can my eye show me clearly both what I see way in the distance, but also close up, and then in the distance, and then also close up? Yes, theoretically. If I have my old man reading glasses on, because I can't see up close without those. But anyway, why can an eye do that? Because when you look in the distance, your ciliary body relaxes and your lens gets thinner. Then when you go to look at something on your phone, now you're closely viewing something, and so the ciliary body contracts, right, and your lens gets thicker. And if I go from reading to back to looking in the distance, I just switched, right? What was contracted is now back to relaxed again. So if literally you keep alternating between looking up close, looking in the distance, looking up close, looking in the distance, your ciliary body keeps switching from relaxed, contracted, changing the shape of that lens. Yes?
So in the module, is that when you were looking at the car, is that moment of you see yellow, and then when you moved it again, it was back to the black dot?
I don't remember that virtual lab so specifically that I can point or remember that exact point in the thing.
Like you were saying, it started off, but as it got closer, it was like a moment where it was like, now you're too close.
Yes.
It was like no dot, and then when you moved it a little closer, the dot was back.
But I think that one was more about the blind spot than it was about the focusing of the lens. Now that I'm hearing what you're describing, I'm pretty sure the virtual lab you're talking about was the one where you're finding your blind spot.
Okay.
So we're going to get to that in a little bit, and then we'll circle back around to that. Okay, great. But hopefully that makes sense, everybody, that we're going to talk again about how we focus for distance versus close-up later when we get to the physiology part. But at least as we're naming parts, I didn't want to just be like, well, here's the ciliary body, there's the lens, without telling you kind of what they do. But hopefully that makes sense that we'll revisit this, right? Your lens focuses light. Think about it. Your lens of your eye is almost similar to the lens you'd find in a magnifying glass, right? So question, if you're looking at something through a magnifying glass, can you hold a magnifying glass anywhere you want and see through it clearly? Or do you have to move it to the right spot to magnify and be clear? Move to the right spot. Well, what if a magnifying glass, when you moved it around, could change the thickness of the glass in the magnifier? Would you then have to move it around or to find the right spot? No, it would be auto-adjusting. That's how our eye works. You don't have to find the right distance to see what you're looking at clearly, because your magnifying glass, that is your lens, changes its thickness to correspond. Hopefully that makes sense as like a silly analogy visual, right? Okay. How are we doing on time? Okay. I often like to give us a little breather in the middle. Let's go for like another 10 minutes, 15 or so, that when we hit about the halfway mark, then we'll take a little, at least 5 minutes to, take a break, and then we'll come back. All right, but we might as well press for just a few more minutes till we hit that halfway mark. All right, so if I'm thinking clearly, we now finished the vascular layer, right? We covered the choroid, ciliary body, the iris. So that officially brings us to the sensory layer, which we can also call the retina. It is within this retina that we're going to find different types of specialized neurons that allow us to absorb the light and eventually send it to the brain for processing. So what I want you to notice first, before we move images, is that inside the eyeball, once you remove the next layer, right, like the vascular layer, and you look inside, in the diagram on the screen, they kind of use a yellowish color for the retina. On this plastic one that I'm holding up, it's kind of like a peachy kind of color. But everything you're seeing in there or there, right, is that retina. What do you notice? Does this retina here come all the way up to the lens, or does it stop somewhere along the way? It stops. What do you think the name ora serrata is telling you about that borderline? Is it smooth or zigzagged? Zigzagged, because it looks serrated, right? As in the word serrata. So when we look inside the plastic model and you see that kind of peachy colored membrane that is the retina, notice when it ends, it does so with what looks like a zigzagging little borderline in there. That would be that ora serrata, right? Serrated edge to the retina. Which begs the question, why does the retina stop there and not go like all the way to the lens? There's a very logical reason why at some point the retina just stops at about there. Can light curve, or can it only travel in straight lines? It can bend when it changes from one medium to another, but when it's in a single medium, how does it travel? Straight lines. Light can be bent by what's called refraction, we'll talk about that soon, but as it travels through any medium, it never curves, right? It always travels as a straight line. That said, can then light ever come into a person's eye and hit here? No. Right, if you think about it, light can enter and hit back here, here. We could do some crazy angles, right, and have light hit like up there. But at some point, there's going to be angles you just can't hit because the light literally can't curve to get there. So would there be any benefit to having any retina here? No, because it'll never be hit by light. Then it's just wasted cellular energy. So that's why it's logical that at some point in a model that shows the inside of the eye, this retina doesn't come all the way up to the lens, it stops. Because everything in front of that are angles at which the light could just never hit anyway. All right, so that's the retina kind of coming to an end at its ora serrata, that zigzagging borderline. All right, but here's the thing. We have to start going to more zoomed in images because this is like macroscopic and a whole bunch of the detail of our retinas is at the microscopic level. So let's go ahead and switch to a more zoomed in view to start looking at that microscopic detail. Okay. So notice, this image to our left is supposed to represent a chunk of the eyeball wall, and it's supposed to be showing us the multiple layers, especially now, of the retina. Something to show you is that on the two-dimensional flat plastic models at each of your tables, there's a rectangular inset picture to the bottom left, and what I want you to notice is that that inset picture looks just like this. In this little piece on your models, notice that at one end of that inset picture is a dark stripe. That dark stripe is this, the pigmented layer of the retina. That's why it looks so dark, right, on the plastic model at your table, because it is heavily pigmented to add to that darkness of the inside of the eye. Whether we then look at the models at your tables or this diagram here, what does it look like? How many layers of neurons does it look like builds the neural layer of the retina? 3. Look at the little rectangular inset picture on these plaques at your tables. There's a brown layer of cells right next to the dark stripe. Then there's an orange layer of cells in the middle. And then there's a yellow layer of cells toward like the nerve building side of the image. Well, what do you know? There are 3 layers of cells, right? Photoreceptors, which are the brown ones, bipolar cells, the orange in the middle, and ganglion cells are the yellow toward the nerve. That's exactly what we're seeing here, right? The first layer near the pigmented are the photoreceptors. Second layer are the ones in the middle, bipolar cells. And then finally, third layer, right, ganglion cells. What part of a cell do we notice most, like usually dark under a slide? The cytoplasm or the nucleus of a cell? What's the darker structure you typically see on a slide? The nucleus. look at how a slide of a retina shows there are 3 layers of neurons, because you literally see 3 rows of clustered nuclei. Right? I see a whole bunch of cell nuclei in a row there. I see a whole bunch in the middle there, then a whole bunch down here. That shows me there's 3 layers of cells in the retina. And because these are all specialized types of neurons, that's why all of that is called the neural layer. Okay, which of those names literally tell you I pick up on light as a stimulus? Photoreceptor. Photo refers to light, and a receptor is a structure in the body, right, that picks up on a stimulus. Right? Thermoreceptors pick up on temperature, right? Photoreceptors pick up on light. Okay, look at these names, rods and cones. Anybody know which of those allow you to see like in black and white, and which allows you to see in color? Yes. Rods are for like black and white, cones are for color. So a good way to remember is the C's stay together. Cones are for color. Okay. Quick question. Do you think rods and cones are evenly distributed in your retina? Or do you think rods and cones are in like specialized areas? Yeah, they're specialized areas. Let me ask you this. Where do you see color the best? When you're in a well-lit space or a really dark space? Well-lit. That's because cones work best in bright light. So where are you going to put more cones? On the extreme edges of the retina or smack in the middle? The middle. Because what hits there more? More light. So interesting, rods and cones, right? Rods allow like black and white, cones allow for color. But are they evenly mixed in your retina? Definitely not. You find more rods on the edges, like your peripheral vision, and lots of cones in the middle, because that's where most light hits. Question, what if a person was born with a genetic mutation in their cones? What would that produce? Color blindness. All right, let's see if anyone's colorblind. What number do you see? 74. I'm sorry if you don't see the 74, you just found out in real time that you're colorblind. Anyone? Anyone? No? That's good, okay. Okay, right, this is literally a standard colorblind test. You hide numbers or letters in specific colored dots, right? Every dot making this number 74 is a shade of green. So what if I had color blindness that did not allow me to see green? Would I see the 74? No. I literally once, like 20-something years ago, had a lab student at a 4-year school where I was teaching, flipping through a book of these, going, I don't know these numbers you all are talking about. An 18-year-old student found out in lab that day that he was colorblind. That happens. I was like, well, you're not going to be a pilot. Anyway, yeah.
So does the green look like gray, or just it all blends into red?
I'm not sure, obviously, coming from like a non-colorblind perspective, and right, different people have different types of color blindness. My mom's partner happens to be colorblind, right? And as I've talked to him about it, he sees things in colors that are not what he sees them as the wrong color. So for example, he and my mom live on like a golf course, and balls just show up in their yard. They collect them and put them in a thing. And he'll be like, today I found a blue one. And my mom's like, that's hysterical because that is bright pink. And he's holding a pink golf ball. We're like, look at this neon blue I found. And we're like, that's hilarious. It is completely not that color. So it's not a gray. They're seeing it as a wrong color. And so therefore, if that wrong color was also scattered around, it becomes then what symbol? I'm not seeing a symbol, like a letter or a number or whatever is supposed to be hidden in the dots. It becomes jumbled for them. Yeah. All right, not that I'm an expert on that, but that's the idea. Anyone know perchance, is it more common to find colorblind males or females?
Males.
Males. Anyone know why? Color blindness is genetic, and the gene for it is on the X chromosome. How many X chromosomes do females have? 2. So that means that because color blindness is recessive, A female needs 2 copies of the gene for colorblindness to have it, because if she only gets 1, then she still has her other X chromosome to hide the recessive disorder. But what about males? How many X chromosomes do males have? 1, because they have an X and a Y. So if that 1 X chromosome he has the disorder for colorblindness, is there a second X chromosome to hide that? No. That's why anything on an X chromosome is seen way more in males than it is in females. So that's true for colorblindness, pattern baldness, muscular dystrophy, where there's a number of X-linked conditions that are generally only like male-oriented disorders. Interesting, if you ever meet a female who is colorblind, what is true of her father? He has to be colorblind, because that's where her only second X chromosome source is. Of course, if you meet a female who's colorblind, you say, oh my gosh, so your dad has to be colorblind. And if she says no, then you're like, I shouldn't have said anything. I'll be out here. It's not your father. But anyway, I digress. Maybe just don't say anything. Yeah. Don't create an episode of Maury Povich in everyday life. Okay, so that is the idea of rods and cones. And let's do one last thing before we take our breather. You correctly told me just a few minutes ago that rods would be at the more edges of the retina and cones more in the middle. That brings up the idea that there are areas in the middle of your retina that are so bright that you load cones there. So for example, this red circle right here is your macula lutea. We can shorten it to just macula. What do you call it in an elderly person who's like 75, 85, where all of that starts to break down? Macular degeneration.
And there's 2 different types, right? So how does that work?
Ooh, I'm not familiar with the subtypes.
Wet type and a dry type. I only know
Ooh.
this, not to be personal, my
Yeah.
dad has it, so that's fine now.
Yeah, and that's what I have to look up.
I'm pretty sure there's 2 different types.
Yeah,
One's
that's-
worse than the other. I don't know enough about it.
Yeah, me neither.
It's actually.
Yeah, right, I know, I'd same thing, I'd be like, hey, ChatGPT, what's the difference between one dry and a... Anywho, right, but the idea generally is, everybody's clearest vision is like right in the middle, because that's where you have this big density of cones in that macula. The problem is, once we pass like 75, 80, right, some people's macula starts to degenerate. And that's where that person's vision starts to get fuzzier and fuzzier in their middle. And so you'll find like some people in their 80s that will be like, don't mind that I'm kind of looking at you with my peripheral vision, because if I look right at you, your face turns to a blur. Because their middle of their vision is just degrading, right? Well, here's what's interesting. There's one little spot in the middle of that macula that gets hit with so much light, then there is only cones in there, literally not a single rod to be found. That little dot, shown as the yellow dot there, is the fovea centralis, but again, very common to drop the second word and just call it the fovea. Okay, so here's the idea. Because the fovea is inside the macula, then on something like the back of this model, where there are little arrows to indicate those structures, it's impossible for me to just put a sticker there and you for just to like know which one I'm asking you, right? So here's the idea. What if I put a sticker back there and I phrase the question saying, name this area with cones only? Did I just describe the macula or the fovea if I specify that there are only cones within it? The fovea. But if I point in that vicinity and say, name this area with mostly cones, which implies that there are still some rods, then what does that have to be? The macula. Right? Your notes specify that the macula is mostly cones but still some rods, whereas the fovea is purely cones. That's my little way of then making sure that if I put a sticker on there, I'm asking, can phrase that to make sure you know which one to answer. All right. So we still have a few structures left to go through of the retina, but this is a good like little breaking point. When we come back, we can finish up the retina and the rest of the notes, and still leave plenty of time to like look at plastic models and our sheep eye specimens. On some lab days in the future, I might say let's take a good 10-minute break, but because today's logistic first day busy, then let's try to keep it to 5. All right, so we'll resume in about 5 minutes. Hey Siri, is there a timer for 5 minutes? She listened to me. All right. Seating chart just to help me with their names, so it's going to be good. Okay, what's your name? Okay, thank you. Nope, my office printer is out of toner, so I screen grab like the roster on my phone and just jotted down names into a seating chart, so kind of attendance. I didn't get your name yet. When you have a chance, every student has to sign the safety contract showing that you know about the lab protocols. Here's one. This is the safety agreement. We have a chance to use it, the place to sign it. Oh, I didn't know you were on this. Can I sign the one? It's not a legally binding document. separate exam or one big exam? Finish off the retina part of today's lesson. Just again to clarify, right, when we look at the zoomed in area called the retina detail, that's where we see a dark stripe, which is supposed to be the pigmented part of the retina. Then we see those 3 layers of neurons. The first layer being the photoreceptors with those rods and cones. What we already discussed them as right, rods are for black and white, cones for color. But now we can point out, well look, the rods are literally long and thin, like a rod. And the cones literally have a pointed shape to their, like pointing like a little tip, like the shape of a cone. So literally on this retina detailed little box, you'll notice that most of the brown cells are long and thin, but every once in a while there's a thicker one in there with a pointy tip. And that would be a cone. The entire middle row of cells, shown in more like an orange color, just like here, are the bipolar cells. Literally given that name because they are bipolar neurons, right? One axon, one dendrite. And then finally, the reason the last name of a row of cells called ganglion cells kind of makes sense is because these are the cells whose axons are bundling together to build the optic nerve. Literally, in the nervous system, a ganglion is like a little bit of gray matter along a nerve. And literally, these are like gray matter structures here, these somas building a nerve. So that's the idea that we see our 3 layers of our neural layer of the retina on that little detail. If we then kind of zoom back out, if you will, we should then realize that this bundle of axons that is the beginning of a nerve is eventually going to come out the back of the eye as the optic nerve. So literally, right, this big nerve coming out the back of the eye is where all the axons of all of those ganglion cells finally bundle together into a cylindrical nerve. Okay. There's a big difference though between my highlighting or putting a sticker on this structure, which is the nerve, versus instead highlighting or putting a sticker right here, and that's where we call it in this diagram the optic disc, but it's also commonly called the blind spot. Why would it make sense that only right here, where the nerve is leaving the eye, is there a blind spot? What do you think? If there is what looks like blood vessels entering and exiting here, and this is also where all those axons are exiting, does that mean that there can actually be rods and cones right there? Or do all those vessels and like nerve axons kind of get in the way of where there would be rods and cones? They get in the way. So literally, there's a circular area at the back of each eye, right in front of the nerve, that is your blind spot. But do you walk around all day being like, well, I see nothing right there, I see nothing right there, and I just have to be okay with that? No. The reason that's not a thing, right, is because whatever's in the blind spot of your right eye, your left eye can see, and vice versa. So therefore, your brain kind of takes information from the opposite eye and fills it in. But even if you close one eye, it's not like the blind spot then appears, right? Well, that's also fixed by our brains, if you will. Even when you have one eye closed, the open eye does tiny little movements and gathers as much information as it can to like fill in the blind spot. What's interesting though is when it does that, it can make mistakes. So that's where one of the virtual labs, as you were referencing before, is where it encourages you to like close one eye and move like relative to your computer screen, and a shape on the screen disappears. Then you move a little bit and it comes back. The moment it disappears, it's not because it left the screen, it's because it moved into your blind spot. And your brain then doesn't even know it's there. Then you move a little bit and it exited your blind spot and it reappears. So that's the idea that one of the virtual labs is like you playing around with an optical illusion, finding your blind spot. Kind of cool. So that is the idea then that everybody's vision, to a certain extent, is your brain's best guess at what you see sometimes, in terms of filling in that little blind spot. Just interesting. All right, so that's the idea then. If I put a sticker like on the actual protruding structure, right, that's the optic nerve. But if instead the sticker's on the inside of the eyeball, like right where the nerve would begin, then it's the blind spot or optic disc. Okay. Quick question. Look at this image here, the top left. Do optic nerves come straight out the back of the eye? Do they go laterally toward the ears? Or do they go medially to crisscross each other? The two optic nerves. Medially. Right? We should have learned in A&P1, your two optic nerves form a cross called a chiasm. Right? They make an X. So what does that mean? If I look at a plastic model, can I use the tilt of that nerve to know if it's a right eye or a left eye? Yes, because an optic nerve always tilts medially. So for example, right now I have this pupil of this model facing perfectly to the back of the room. But as I spin the model around, the nerve is not perfectly like straight to the ceiling, it's tilted this way. So is this a right eye or a left eye if this nerve right now is going like this? It's a right one. Because if I put it to the left, the nerve is then angling out toward my ear, which makes no sense, and this is a right eye. So notice, does the tendon on top only reinforce that interpretation of this being a right eye? Yes, because the tendon is also going toward the midline. So we've learned 3 different ways to tell a right eye from a left eye. One is the gland for making tears, which is always laterally positioned. Then there's the tendon for the oblique muscle that faces the nose. And then there's the nerve coming out the back that tilts toward the nose. So plenty of ways on any model that you should be able to orient yourself. And again, I might never ask right or left, but if I ask what muscle is this, the only way if you know that's medial or lateral is if you know if it's right or left. All right, excellent. So that is the idea of the optic nerve and the corresponding optic disc. And so with that, that should finish off that topic there of the retina, which finally then brings us to Roman numeral 3, all about the lens, the chambers, and the fluids. So we jumped ahead, I'm just actually on this picture, we already jumped ahead to the lens, right? Because we looked at it earlier in the context of the ciliary body. But now you finally get notes that explain that your lens is a biconvex, transparent, flexible disc. Biconvex. The opposite of that would be biconcave. What have you theoretically learned this week is an example of a cell that is biconcave? Red blood cells. What does biconcave mean? It means indented on both the top and the bottom, right? Biconvex means bowing outward on both sides. So the shape of your lens in your eye is like that. Biconvex, bowing out, as opposed to like a red blood cell, right, that goes in on its 2 sides. Transparent, what does that imply? It's clear, right? Light can go through it. But what is the light going to do when it hits a curved lens like this? It bends, right? We said light can't curve, but when light goes through a substance of a new medium, like with a new density, for example, the light bends. That's the idea that the lens focuses light. It takes light that was separate and brings it to like a focal point. Okay, and why does it have to be flexible? Does the amount of bending that you have to do to the light to bring it to a focal point, is it always the same? Or does that amount of bending change whether you're looking at something close up or something in the distance? It changes. And that's why our ciliary muscle has to control those changes, right? Now, what generally happens to the flexibility of any body structure as we age? Does it become more flexible or more rigid as we age? More rigid. Which is why I need these, right? I just realized yesterday that I forget how old I am. I'm that old. I was like, wait, am I 48 or 47?
I told us 46 last semester.
So then I'm 47. Oh no, then I'm 46 still? I don't even know. It's like I'm just old enough. Yesterday, my first class of the semester, I mentioned like, oh, 47 or 40, I don't remember, late 40s. And a student was like, really? I thought you were 35. And I was like, that is how you get an A, everyone. Take notes, right? Anyway. Okay, so, right, when I hit 40, all of a sudden my husband was like, do you realize you're holding your phone at your hip? Because I'd be like looking at my phone like this. And I'd be like, I am? And he's like, look at you right now. I'm like, this. And so, of course, right, this becomes a reality at some point. The idea is when you're very young, like children, everybody has what's called a near point, meaning the distance from your pupil that you can see clearly. Well, 10-year-olds and younger have a near point of only 5 centimeters. Like meaning a little kid can pick up like a little book and be like, I'm reading, and hold it almost touching their nose, and they see it completely clearly. And their parents are like, what are you doing? You're gonna hurt your eyes, hold it here. Because their parents literally now have a near point that is like 2 feet, right? And the grandparents are like, how can you read that? Hold it across the room, right? With a candle, with a flashlight. But the reason for that is that as we get older and older and older, that distance becomes further and further. Because in order to see up close, right, you have to make the lens get really thick. Well, at some point, your lens just resists getting thicker. And that's why your near point keeps getting further away. That's why at some point, you got a magnet. I have these like from Amazon in like a 10-pack in a box. Because when I like break a pair, it's like on to the next one, take toilet paper. Just roll them over to the next pair. Anyway, I'm oversharing, that's what I do. Right? But that's the idea, right, that I'm already on like my 2nd or 3rd level of prescription, right, where they keep getting like more intense, right? So you've all got this look to look forward to. It's inevitable. Anyway. All right, that's called presbyopia, right? The inevitable need of magnifiers, but regardless, that is the idea of the lens, right? Focus is light, but unfortunately gets more rigid as we age. Okay. Your lens is a distinct borderline in the eyeball. If you're in front of the lens, you're in a space called the anterior segment, anterior chamber, anterior cavity. Doesn't matter as long as the word anterior is in there. Okay, so what's behind the lens? Posterior segment, or cavity, or chamber, whatever you want to call it. The thing though is, there's a big difference on an exam day between my putting a sticker in those spaces and saying, name this space, versus name the fluid that fills this space. In the front of the lens, there is a fluid called aqueous humor, and behind the lens is a fluid called vitreous humor. Which of those makes it sound like it is watery? Aqueous. Which one sounds like jelly? Vitreous. Right, the word viscous, right, implies a thicker fluid. So vitreous is like a thick jelly, whereas aqueous is more water-like. One of those two has to last you your entire lifetime because it never turns over, and the other is constantly turning over like cerebrospinal fluid. Which do you think is which? Which has to last you a lifetime and which constantly turns over? It's hard to make new jelly, right? Once you make a Jello mold, right, you can't like add to it, right? It's there. But if something is based in water, can that turn over from your bloodstream? Yes. Because our blood is mostly water and aqueous humor is watery, it can leak from your bloodstream and turn over the same way CSF does. But once you make a mass of a jelly in the body, you can't like turn that over. It's there permanently. So that's the idea that when we open up the plastic model, the only fluid they choose to represent is the jelly-like one that is like this big thing filling the back of the eye. If I were to ever ask you what fluid does this represent, that's the vitreous humor, and notice it's sitting behind the lens. Okay. They don't actually, on the plastic models, represent the aqueous humor, but if I put a sticker here in the gap of space in front of the lens, and I say what fluid fills there, right, that would be the aqueous humor. So again, the name of the spaces are the posterior and anterior segments or chambers or cavities. But the fluids, right, are vitreous humor, aqueous humor. Okay. Have you ever noticed floaters in your field of view? Right? Especially if you look at like a white wall, right? You might notice like a little speck or like a little black spot, or maybe even a little squiggle, right? Like an imperfection right in your field of view. What happens if you try to look at that little floater? Will it stay put so you can stare right at it, or does it like run away when you try to look at it? Yeah. Why is that? Because that little floater imperfection in your vision is actually a little imperfection in this jelly. What that means is, if there's a speck sitting in the middle of this jelly, that speck, when the light hits it, casts a shadow. So the floater you see is a shadow, not an object. So question, when you then move your eye to look at that floater, does the shadow stay, or does it move because you just changed your angle to the light? It moves. So a floater running away from your eye when you try to look at it is because you're trying to chase a shadow that you can't chase. Because the shadow is cast from the jelly inside your eye, and every time you try to look at that floater, the little floater's like, well, now I'm over here, and now I'm over there. Right? Anyway.
Now, will the floater like go away, or is it just kind of like hanging out in there, and like it just depends on like
Good question.
how the light hits?
So I'll start with this question. Between a person who has only ever jogged athletically as like a sport in their life, like running, versus a person who has played football, lacrosse, wrestling, who's going to, as an adult, have more floaters?
More sport.
The person who's got more head injuries, right? If you're playing contact sports and you're getting like knocked around all the time, is that going to add more imperfections into this jelly? Yes. So football players, boxers, etc., will be like, I'm only 30-something, and my vision is just filled with spots. And it's like, well, keep boxing, that'll help. I digress. I'm just not like a fighting person. Shocker. Anyway. Anywho, I'm like, sport-wise, volleyball. No contact, maybe some tennis. I digress. Okay. Anywho, right, so that's the idea that people that have more head injuries, etc., like have more floaters. Why would it be possible for over time the floaters though to decrease? Well, because what if there is a mass of like protein, cell debris sitting in that jelly? If it's what could happen to that thing causing the floater if it's heavy enough? It can sink through the jelly. Right? Much like if you make a jello mold with a heavy piece of fruit in it, with enough time, it might start to like sink within the jello just by gravity, right? So that's where, yes, they can go away. If we're talking about, let's say, a piece of protein chunk that starts to drift down, that's how it can go away. But that's the only way. Otherwise, it's there indefinitely.
So, sorry.
Oh, please, yeah.
So does it go away and it just like stays at the bottom of your eye? Okay.
Theoretically, yes, right? In most people, those floaters are so small, they stay put. But only when you might have like a boxer or somebody who had a bad car accident, where now there's like a bigger floater that is heavy enough to start to sink through the jelly. And then yes, it would just be at the bottom of it then.
Okay.
Yeah. But in general, most of them stay put. All right, so that's everything to know there for like the fluids and the spaces that are filled with those fluids, which finally brings us to our last Roman numeral, which is then all about physiology. Okay. So first thing we have to learn is a couple of terms. Your retina does something called phototransduction. Literally, it absorbs light as a stimulus and uses that light to trigger the formation of nerve impulses, right, otherwise called action potentials. Your textbook goes through phototransduction over multiple pages, light reactions, dark reactions, the different proteins involved. It's a lot of biochemistry, and we are doing none of it. So we just know the definition of phototransduction. We're not doing all the biochemistry of it. Right? If you ever decide in the future to become an ophthalmologist or something, I'm sure you'll learn it there. That said, we have to understand some basic physics in biology quite often. So, for example, today we have to talk about the physics of light. Next week we have to talk about the physics of sound. In A&P 1, you have to even learn some basic like physics/chemistry when it came to things like action potentials, etc. Right? So physics is an important part of biology. So for example, light has a property known as reflection. Quite literally, it's the ability of light to bounce off of an object. Right? You turn on a flashlight. If you shine that flashlight at something that has a bit of a reflective nature to it, right, that flashlight will bounce off the wall, right, and now be shining almost back at you, right? And that's a property of light, that it has the ability to bounce off of objects, and we call that reflection. Quick question. When you see something as a specific color, like red, is that because the light is being absorbed as red, or the light bouncing off is red? Bouncing off. The color you see is the color reflected back at you. So when you see an object as red, that means that object is absorbing blue, green, right, the other colors of the spectrum. What color is reflecting off the object and hitting your eye? The color you observe. So that's the idea that light can reflect, bounce off of things. And very often, the full spectrum of white light, many of the colors are absorbed, and only one color reflects, and that's the color you see the object to be. Okay. But, even though we already said earlier that light can't curve, light, however, can bend. But it only bends when it leaves one substance and goes into another substance, especially when that new substance is a different, density. So for example, notice how this spoon, between the spoon and our eyes up here is just air. Okay, between the spoon and our eyes here is glass and air. Okay, between the spoon and our eyes down here is water, glass, and air. Which means that there are specific spots where there are transitions, right? And so therefore, is the spoon actually broken in these places, or is that just an illusion from the bending of the light? It's an illusion, right? You probably noticed if you've looked into a fish tank from one angle, then you look at it like the side of the tank, and the fish you thought was in one position now looks like it's in a totally new spot. And it's because you're looking through different amounts of water, and the glass is bending the light, right? It creates a bit of an optical illusion. Similarly, if you ever looked at a picture of somebody's backyard that has a pool in it, in almost every photo, a pool looks 2 feet deep, even if it's 10. It's an optical illusion of how the light bends and stuff. So that's the idea that light bends, especially when light moves from water to air, glass to water, right? Well, let's think about everything that light passes through before it gets to your retina. Okay, light is hitting your eye. What's the first thing it goes through? What's the clear window on the front of the eye? The cornea. Then it goes through what fluid filling the anterior segment? Aqueous humor. Then it goes through what structure? The lens, good. And then finally goes through what fluid? Vitreous humor before finally hitting that retina. What is the light doing every time it goes air to cornea, cornea to aqueous, aqueous to lens, lens to vitreous? Bend, 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 bend. Which is the only piece of that is adjustable, how much you bend the light? The lens. So the idea of refraction, the property of light to be able to be bent when it transitions from one substance to another, is actually utilized by our eyes, right? The light bends every time it transitions from cornea to fluid to lens and so on, but there's one place where we can control how much that bending is, and it's the lens, because we can make that lens thicker or thinner. All right, so that is the idea of how we can utilize the properties of light, like reflection and refraction, to see the world around us, hopefully in clarity as well. Okay, so then the question is, how do we focus for close-up vision versus, say, more distant vision? So let me ask you this, which one leads to eye strain? Imagine you spend 3 hours of your day driving where the entire time you're looking like miles off into the distance, and then you get home and spend 3 hours reading with no breaks. During which will your eyes be like, okay, my muscles are just achy, my eyes are like strained. What would it be, the driving or the reading for 3 hours? Reading. Why? Because looking way off in the distance, your ciliary body's relaxed. And why is that? We can start on this picture. Let's imagine you're looking at mountains like miles and miles away. The reason you see those mountains is because light has hit the mountains, bounced off the mountains, traveled the miles that it had to get to your eyes and into your retina. That light traveling that many miles must have been coming in parallel, because that's the only way it hit your retina from that many miles away. Okay, but what if instead of looking at miles away, I bring a piece of paper in front of my face? Is this light coming at my eyes in perfect parallel, or is the light like diverging, scattering in my face? If it's like light bouncing off right here, scattered. When you're looking at something in the distance, all the light scattering doesn't enter your eyes because it had to travel such a far distance. Only light in parallel goes into your pupil. When you're looking at something close up, more of that scattered light can enter. That's why it's harder to focus up close, because the light entering is more divergent. When looking at something in the distance, the light had to travel so far that it comes in parallel, and therefore it's easier to focus. So, look at how this person's looking at mountains. It tells you the ciliary muscles relaxed, and look at that lens, it is flat. This person could do this for hours because there's no muscle work being done. But then this person's like, well, look at the time, apparently, as they like look at their watch or their phone. This is now close to their face, right? What is the case now? The ciliary muscles contracted. What does that do to the lens? It's now thicker. But will this muscle get tired if it has to hold this contraction? Yes. So literally, I put on your handout that focusing in the distance is like easier, because the light coming into your eyes in parallel, so it is like easier to focus. So that's why the ciliary muscle is relaxed and the lens is flat. But notice it then says for letter E, focusing for close vision, those light rays are more divergent as they enter the eye. And that's why you have to do the accommodation of the lens, event number 1. The ciliary muscles contract, and that makes the lens bulge. But is that accommodating of the lens the only muscle work your eyes have to do for close-up vision? Nope. What do your pupils do? Do they like dilate to let more of that divergent light in, or constrict to limit how divergent the light is? Constrict. Is that a second muscle having to hold yet another contraction? Yep. And if you're looking at something really close to your face, are your two pupils going to be perfectly parallel, or do they have to be a little converged for that close-up vision? A little converged. Is it comfortable to keep yourself in a kind of cross-eyed position for very long? No. So this is just begging for a question that's like, you go from looking in the distance to looking at your phone. Which of the following just changed? Did your lens just go from thick to thin? Thin to thick? Did your pupil just dilate or constrict? Right? You should know that if I just switch to looking something up close, that means my pupil got smaller, my eyes got a little cross-eyed, and my ciliary body contracted. If I look in the distance, then all those things relax, right? So that's the idea that if I ask you about close-up vision versus distance vision, right, you should know which causes strain and why. All right. Finally, the last topic of the day is understanding the need for eyeglasses. Not reading glasses, right, but like prescription eyeglasses. Here's the idea there. If somebody has normal vision, that means the shape of their eyeball is like just right, so that way when the lens focuses the light, The focal point is perfectly on the retina and you get clear vision. However, your eyeball may be a little bit too short, or your eyeball may be a little too oblong, like football shaped. And when that happens, your lens doesn't know the shape of the eyeball. It just bends however it thinks it's supposed to bend. But that might mean that your focal point is too far back or too far forward. And that's when a person can be either far-sighted, hyperopic, or nearsighted, myopic. Right? The idea is when somebody has myopia, nearsightedness, right, everything they see right in front of them, like reading, is fine. But they go to drive and they're like, I can't read those signs, right? And the reverse is true for hyperopia. Those individuals can drive and see things off in the distance, but the second they go to read a book, it's like, oh no, need my glasses, right? Interesting. How then do lenses of eyeglasses work? What is their job? Notice how the focal point in the untreated eye is not touching the retina. But when you put a lens of eyeglasses in front of the eye, what has happened? It's now on the retina. Because what did the lens in the eyeglasses do to the light? Bends it. Is this eyeglass lens trying to change the bending of the light so that therefore the actual eye lens will no longer put the focal point off the retina but now on the retina? Yes. And that's literally what an optometrist's job is, to be like, tell me when it's clearer, with this lens or with this lens, right? And they slide them in and out. And the idea is that the optometrist is trying to figure out how much do we have to bend the light through your glasses to make up for the difference that your lens is not focusing quite right. Can that change over time? Can a person's prescription be adjusted? Yes, right? As you age, the shape of your skull may change a little bit, or the connective tissue behind the eye might get a little tougher, and that can change the shape of your eye little bits over your aging. That's why a person's prescription may change, right? But that's the idea, is that literally it's the shape of your eyeball that determines whether you're nearsighted or farsighted or not, right? That you have, clear vision. And that's the idea then of why eyeglass lenses are meant to bend the light accordingly. Not that I would test you on this, but what do you notice about the shape of a corrective lens for farsighted versus nearsighted? One's convex, one's concave. That should make sense because one is moving the focal point forward and the other is moving the focal point backward. So the curvature of the lens switches for the two different disorders. And not that I would test you on this, but what if two people are both nearsighted and you look at each of their eyeglasses and one has thicker lenses and one has thinner lenses? Who has the more extreme nearsightedness? Why does it have to be the person with the thicker lenses? Because the thicker lens means you have to bend the light more to accommodate for how badly off, right, their lens is doing the focusing, right? A thicker lens means more bending because that's a bigger disorder. So that's the idea. All right, so here's the thing. Well, I still have your attention up at the board here for just a couple more minutes. I'd like to bring us into Connect for just a couple of moments, show you what our virtual cadaver eye looks like, as well as maybe a couple of slides. So just to show you, I give you literal click-by-click instructions as to how to go into the Connect apps. So feel free to go by those should you need to help you navigate. But simply enough, I'm going to bring us out of Brightspace and into Connect, where I can open the virtual cadaver app so we can look for a few minutes at the virtual cadaver's eyeball and a couple of slides. Okay. So when you go into our Connect page, notice this link right here that says Anatomy and Physiology Revealed. That is how you enter the virtual cadaver app. Okay. For those of you that had me for A&P 1, you might remember that last semester I gave you a code to plug into this app. Well, now that you're in A&P2, it's time to add a new code. So in Brightspace, in every lab module, I provide you the code, which for A&P2 you can see right here. Lowercase w, capital F, number 5, then capital J, capital F. If you add that as a list into this app, then this app will not show you any A&P1 content. It will not show you anything that's not covered in this course. It will only show you stuff from A&P2. It's like customizing the app specific to this class. So I encourage you to add that code into this app. I don't have to add it because I've already put it in. I just have to select it in my app. So for example, I just chose the A&P2 code. Look what happens when I go to try and look at tissues, which is an A&P1 topic. It tells me there's nothing there, because I put in the code for A&P2, right? But the I is part of the nervous system. So I choose nervous system, and then sure enough, I'm able to choose vision, and that allows me to literally look at the eye. Well, what do you know? That brings up the virtual cadaver eye, and any name that I click on here will automatically highlight for me in the image. So for example, what did we say that already was the white part of the eye? The sclera, very good. When I click on the name sclera, it will automatically highlight that outermost white layer. And in this case, it also automatically jumped to a sliced view. For example, what do you think that structure is right there with the little white strings above and below? There's the lens. Watch what happens if I click the word lens. Oh, look at that. It just highlighted the lens. So imagine on next week's quiz, I screen grab this with the lens highlighted, and one of the questions on the quiz asks you, the structure highlighted in the image above is blank. And maybe you're filling it in lens, or maybe there's multiple choices, right? But the idea is, notice how you were able to correctly tell me that the white was the sclera, and this is the lens, even though you've never seen this image before. And you were able to do that because we've spent the last hour and a half learning the anatomy, and you're able to then apply that right to the cadaver. Just to show some more, what would this person seems to have brown eyes. What part of the eye is that brown ring of color right there? There's the iris. Watch, if I highlight or if I click on the word iris, it literally highlights the iris. And what is this dark dot in the middle of that iris? The pupil, exactly, right? And I can highlight that. Quick question. Which muscles are these? Notice both of the muscles that I'm currently pointing at are below the eye. But this one seems to approach the eye in a straight line, and this one on a diagonal. So which of these is inferior rectus muscle, and which is inferior oblique muscle? Which is this one? Excellent. Notice if I click inferior oblique on this list, if I can see it. Oh, that's right. It's not on the list because I have to choose the others. Sorry, I have to bring in the muscles. But look at that, you were correct, right? That was definitely inferior rectus, and therefore the one above it, right, is the inferior oblique. So, right, imagine on next week's quiz you see this screen grab and it asks you to name this muscle. Hopefully you would be able to recognize it. Okay. So now that we've seen some structures there, oh, you know what, let me do this. Quick question. What if on next week's quiz you saw this highlighted image and the question said, name the fluid filling the highlighted space? Vitreous humor, good. Notice in this app, it is saying that what's highlighted right now is the posterior cavity, but that's the name of the space, right? But what I want to point out, which is great with this app, there's an info button. I currently have the posterior cavity highlighted, And if I click the info button, it tells me all about the posterior cavity. For example, it is filled or occupied by the vitreous humor. Right? So there's always little background info should you want to read it. But once we're done looking here, like at the parts of the eye that we can highlight, right? For example, oh, look at this. Optic disc versus optic nerve, right? Blind spot versus the nerve that actually transmit the impulses.
If you said blind spot, you'll take that.
I would, yes. All right, let's switch to slides for a second. Tell me, this slide of the eye, is this eye facing the back of the room or facing the hallway? Hallway. How did you know? Because this looks like the cornea side, right? And that looks more like the nerve. What's that big red thing gotta be? There's the lens, right? Separating anterior segment filled with aqueous from posterior segment, right? Look at how this is the very first time you're seeing this image and you're calling out which way it's facing, what's gotta be what, right? Because it's not about memorizing images, it's about knowing the anatomy, right? So, of course, just to show you, you were correct, right? Cornea is to the front. Yes, that was the lens, right? The nerve is in the back, right? You were correctly orienting yourself to this new slide, which is great. Let's look at a slide of a retina. I'm going to go ahead and highlight the pigmented layer, which is right there. How many cell layers did we say built the neural layer? Oh yeah, 3. And what do you know? 3 rows of nuclei. Literally, photoreceptors, bipolar cells, ganglion cells, just like the 3 rows of cells in the little inset on our plaque. And one last slide to show you. Kind of cool. This is a crazy, powerful microscope's zoomed in view of rods and cones. What do you think all these skinny ones are? The rods. But what about like a thick one like right there? There's a cone. Right? There's another one, right? Pretty cool. This is called a scanning electron microscope that almost gives you like 3D-ish kind of view of a tissue. So notice how all the rods are skinny, whereas those cones, right, are the thicker ones. Kind of neat. All right. So here is the deal. That's pretty much everything I wanted to show you from the virtual cadaver app. So what I'm now going to do is throw on a pair of gloves, and we're going to dissect a sheep eye. Good times. All right.